Welcome to the first POCDX seminar this semester. Today we have Dr. Holger Becker, who's the co-founder and CSO of ChipShop in Germany. He did his undergrad at University of Western Australia in physics and his PhD at University of Heidelberg. Working on miniaturized systems for chemical analysis. And he's the chair of SPIE, editorial board of the Lab on a Chip journal and physics journal. So he'll give a talk today. One announcement, the seminars will be moved permanently to Tuesdays for the semester at this time. Next week we have a special seminar with Dr. Dino De Carlos at 3 p.m. in the afternoon, so it's a different time.
All right. Thanks, Amy, for having me here. I will talk a little bit about microfluidics as an enabling technology for point of care from an industrial perspective. So you will hear a couple of things which might be a little bit painful for students. So we'll see. I will first talk a little bit about the, let's say, commercial world of microfluidics, where we are right now. And then I will talk a little bit about strategies of developing point-of-care diagnostic devices, cartridges. I will show you 2 or 3 examples, and in the end, if I might have a few more minutes, depending on how well it goes, I will talk a little bit about the company, who we are and what we're doing. Because this is a sort of fairly informal seminar. If you have any questions in between, just raise your hand, shout at me or so. I try to answer as best as I can. So as a motivation, microfluidics has been around for quite a long time now. The big question is, why did it take so long to become an integral part of product development in the life sciences or in diagnostics? Because if you look at the early papers, the big, let's say the fundamental paper of Andreas Manns, where he explained the concept of a miniaturized total analytical system, that was 1990. So that's 25 years ago this year. And it's still, if you go to a doctor's office or to a hospital, you will not find too many microfluidics-enabled diagnostic devices there. So the big question is, where does that come from? And there are a few reasons to that. If we do basically a reality check, at the moment, of course, we all pride ourselves to say we do lab on a chip technology. But what we do more in reality, I think you see that picture should be fairly familiar in the sense that, of course, it's a situation where it's more a chip in a lab. So you have a small microfluidic device, but you have a fairly complex setup around it with pumps and valves and flow controllers and stuff like that. If you look at the protocol side of it, very often people do many steps, especially things like sample prep off chip, because it's just so more convenient to do that in an Eppendorf or a 96-volt plate, and only do one specific thing on a chip, which of course is not what you would like to have for a commercial solution. For a commercial solution, you really need something which is sample in, push a button, go drink a coffee, result out. And of course, in the past, the development has very often been technology driven. People have been doing things because they just had a mask aligner or stuff like that sitting around and not application driven. And that is always a critical thing. And in addition, and that's now more from the industrial perspective, microfluidics is a truly multidisciplinary field. And if you miss one of these things here, it just won't work. So you have to know the physics of microfluidics, of course you have to understand, you have to calculate back pressures and stuff like that. And of course you have to have application know-how depending on which field this whole thing should be working. Then from, again, the commercial perspective, the thing which most often is not working is system engineering. People very often don't really have that holistic system understanding. And I will come back to that a couple of times during my talk. Material science is a big issue. You have to understand things like surface interactions, optical properties, things like how do proteins attach to surfaces and things like that, microfabrication technologies, etc., etc. So, and if one of these pieces in the puzzle is missing, the chance that the device is working is dramatically reduced. Secondly, again from the commercial perspective, microfluidics always had a very, very high activation threshold. Imagine you're an engineer in a biotech company or pharmaceutical or diagnostic company and you think, oh yeah, well, we've been running this ELISA or so in a 96-volt plate for the last decade. Maybe it's a good idea to put that onto a microfluidic platform. How do I start? You scratch your head, you might start to draw up your design. You have to find a company to make this device for you. The probability that the first device is working, the first design is working is basically 0. And then imagine under these conditions you go to your boss and say, you know, please give me $50,000 in half a year's time, but I will tell you that this thing is not going to work, so I will be coming back in another half year time and ask for the same amount of money. This is a very difficult psychological proposition, so there has been always a very high activation threshold to get into the use of microfluidics. And that actually is still the case, and I will show you one of our approaches, how we try to resolve that situation. And finally, of course, there's this famous Maslow's law of the instrument, formulated in 1966. If all you have is a hammer, everything looks like a nail. Which basically means, and that is a little bit of academics bashing, that even in academic fields, it's very difficult. The adoption rate of new technologies is much lower than people anticipate. So, you know, in biology, for example, people have been doing cell culture in petri dishes for decades. So, in their mind, you know, if all you have is a petri dish, the world is a flat 2D cell culture. And it's very difficult to get people to move away from that. There's a perfect example in the history of microfluidics, that's centrifugal microfluidics, lab on a disk. Has been, I mean, by the year 2000, you had 3 companies out there which had a disposable, a lab on a CD, an instrument to run it, and a set of applications with it. Everything worked smoothly, and I mean, centrifugal microfluidics is very elegant. It can solve certain problems in a very, very elegant way. No pumping, I mean, centrifugal forces are really powerful. Everything was there, nobody wanted to buy it. Because at that time, the companies had quite heavily invested into lab automation and robotic systems, and now suddenly somebody comes and says, you know, throw away this $250 million investment. I have the next best thing, which is something which turns. Nobody wanted to buy that stuff. And out of the 3 companies who had systems out there at that time, there's only one survivor, which is Gyros in Sweden, which at that time were the biggest life science investment in Sweden. And they are just barely hanging on. I mean, there's like 20 people or so after 15 years. So clearly, a good example for the slow adoption rate of, or let's say, the psychological threshold to adopt new technologies. And of course, microfluidics could be a very nice extension of that toolbox. So in theory, you know, we have very controlled environments. The dimensions we work in is typically very compatible with many things in the life sciences, the size of cells or bacteria or stuff like that. We can do 2- or 3-dimensional structuring. We can do any sort of surface functionalities. We can do the functional integration of fairly complex workflow into a single device. So there are lots of reasons why it makes sense to use microfluidics. And yes, it looks like the markets are finally taking off. This is a fairly recent market study from a French company, Yole, which I think have somewhat of an insight in the microfluidics market. You can always argue about the absolute numbers here. But from my perspective and my experience, I would agree with the way of how that growth rate is accelerating. So I think we can by now say we are living in interesting times in the sense that, yes, microfluidics is becoming an integral part of various markets, mainly in the life science. You can see actually the 2 lower ones are clinical and veterinary diagnostics and point of care. So do we have a pointer or a stick? So these 2 columns here. So I think the growth rates are really nice, and especially they are robust in the sense that we haven't really seen any downturn or so despite any sort of global development of global economies. So when we look at the current state of the market, oh wow. It works? Oh great. So as I said, there's a high entry barrier to use microfluidics, and I already give you a hint of what I will be talking about next in the way of how to lower that barrier, and that is modularization. There are no killer apps around at the moment. The markets are extremely segmented. If you look back, in the early days of microfluidics, everybody was thinking about the killer app, the single product which makes hundreds of millions of dollars in revenue. This has not materialized. Instead of that single product which makes $500 million in revenue, we have 250 different products which make $1 to $2 million each. So it's a very segmented market, and that's the reason also why this market is fairly geared or fairly dominated by small and medium-sized companies. Because if you are, GE or Toshiba or Siemens or one of these big guys, for them it only makes sense to go into a market if they have a business proposition which tells them we can make $100 million or $200 million within 2, 3, 4 years. And microfluidics at the moment clearly is not at that stage. It's very segmented. Again, microfluidics is an enabling technology. 15 years ago, companies were selling themselves as a microfluidic company. Caliper started at one point to say something like, no, microfluidics inside, caliper inside. Clearly this is not the case. Microfluidics helps to solve a problem, but it's not a product in itself. Application know-how is really the key, so we have to, and I think the market actually, or the technology has made that transition from being technology-driven to application-driven. But then, as a microfluidics company, you have to understand the applications. And then finally, and that sort of alludes back to that psychological threshold. I always tell people, if you have a disruptive technology, and very often microfluidics is part of disruptive technology, put it in a conventional box and don't talk about it. So when we see now, for example, all these companies who do droplet-based stuff or so, I tell them, don't tell anyone that you're doing that. Put a conventional box around that and just tell people this is doing things much faster, but in the same way as you've been doing that before. And again, a story out of the situation at the moment. There are two companies around which at the moment offer droplet-based digital PCR systems, Quantalife, or Biorad now, and Raindance. Raindance had about the most amazing business plan I've ever seen. I mean, they came to us some like 15 years ago with thick leather-bound volumes, 3 Nobel Prize winners on their board and stuff like that. They have an instrument which totally displays that disruptiveness. A very expensive, disposable, really, really fancy. They raised $100 million. I'm not so sure if they ever will get that money back. On the other hand, Biorad, formerly Quantalife, spin-off from Lawrence Livermore, started in 2007, was sold to Biorad 2012 for $170 million. They have a fairly simple droplet generator, which generates droplets, and that the droplets then go to a standard well plate-like structure, and it's read out by a standard well plate reader. And I think that is a really smart approach, because the user doesn't have to deal anything with, complex droplet generation or something like that. He can use his existing readers, and that's fine. So that's a good example for not displaying disruptive technology. Because the user is afraid. The inventors are totally in love with that technology, but the users are afraid of disruptive technologies. And so a little bit on value creation, because that shows, I think, the sort of status of the market. We have seen quite a bit of mergers and activities in the last 3-4 years. Caliper is a really good example. I mean, being one of the technology pioneers in the field, the sort of technology showcase and success story for a long time, but clearly not an economic success for a long time. In 2011, they made around maybe $50 million on their microfluidics stuff. Having been on the market for 15 years, that's not a huge figure. And at that point in time, they had something like $250 million accumulated deficits in their books. They were just about, let's say, operationally profitable, but it's on the economic side, not a true success story. But then Perkin-Elmer came and bought them for $600 million. Whoa, suddenly, obviously, somebody is associating value, economic value with technology. The same as I said with Quantalife. I mean, from 0 to 170 million in 5 years, I think that's not bad. And another case, Illumina bought Advanced Liquid Logic, a company doing electrowetting technology for about $100 million, and that's another good case. At that point, Advanced Liquid Logic had been around for almost 10 years, and they were looking, or they had the idea we build a diagnostic product using electrowetting, again, droplet-based something. They never could hit the price point for a diagnostic disposable, because they had to do they had a PCB board for the electrodes, they had to put a cover lid on top. So they were struggling. Now Illumina came and bought them because they were looking for a technology for sample prep for sequencing. And it's of course a totally different proposition, value proposition, if you have an application which is worth $1,000 as a sequencing run, and then you spend $10 on a disposable for the sample prep, or if you have a diagnostic case where that application is worth $10, so you can only spend $1 or $2 on the disposable. So that was a good case. And then finally, another example here from the Bay Area, Fluidigm. I mean, they peaked here in early 2014 in their market cap at $1.2 billion at a time when they were making something like $70 million in revenue and had about that amount of, being far from profitability in that way. So, that's something which I'm always sort of scratching my head a little bit, says, How can you justify a valuation like that? But obviously somebody does. Okay, and finally, I think one of the reasons why we see that upturn in our uptake of microfluidics technology in the market is that we can really fulfill one of the very early promises, and that is the integration of complex protocols, of complex functionalities into a single device. 10, 15 years ago, people have been talking about something like that. Nowadays, we can actually make it. So, the question is, how do I get to a microfluidics-enabled product? If we look at, let's say, a typical molecular diagnostic assay, you can draw these 6 boxes, the 6 steps to happiness, and it's always very easy to draw boxes. So the question is, where's the problem? So you start with a sample interface. Sounds trivial, but it's not. How do I get my sample? That could be blood, that could be sputum, that could be urine, something like that, onto a chip. You then very often, or all the time, have to do sample prep, and again, that is can be a sequence of a number of protocol steps, which might be fairly complex. The problem is this field academically is totally unsexy. This will not give you a Nature paper if you solve one of these steps, but for a real product, this is totally indispensable. This is really, really important. So at that point, you typically generate your nucleic acids, for example, when we talk about molecular diagnostic assay. You then typically have an amplification step. Could be classical thermocycling PCR, could be isothermal, could be continuous flow. By now, there are dozens of amplification technologies available on the chip. You then very often have a separation step involved, so it could be electrophoresis, could be chromatography, could be hybridization. That's actually something which becomes more important. You can use forces, electrophoresis, centrifugal forces, magnetic forces, whatsoever, to pull out the analyte of interest. And then you have your detection. Again, historically, that basically all the time was optical, like fluorescence, for example. We see a big trend towards non-optical detection technologies, especially if you think about something like very small, portable instruments in the way that people tend to think it's easier to read out an electrical signal compared to an optical signal if it's not something like in a pregnancy test, just a color change reaction, which you can read out of the smartphone. And then also something which sounds trivial, but it's not. Very often, especially if you have an infectious sample, you have to contain everything on your cartridge. And just by the sheer volume which you might have in your waste container, we have devices where the waste container takes up about 1/3 of the overall real estate of a cartridge. So again, that is not trivial. But as I said, you can draw these boxes quite easily, and the question is, where's the problem? The challenges here in the development is that the classical engineering approach, if you have a complex problem, is of course deconstructivistic. You chop it up into small individual pieces, solve the pieces, and then you do a linear extrapolation of that. This unfortunately does not work so well in microfluidics, unlike in microelectronics. Because there is a complex interaction of the individual elements. In microelectronics, a friend of mine used to say, the transistor doesn't care for where the electron comes from, which drives the gate. In microfluidics, however, of course, if you have mixers or splitting teas or something like that, they all influence each other, so there is an interaction between the different elements. In addition, there are limits to simulation modeling. That's a good example of something which you can model. That's a comparatively simple structure, or let's say it sounds like a trivial problem. You have one inlet, you want to split up your flow into different chambers. In this case, we wanted to do an organ on a chip, cell culture in these 2 chambers, and then the stuff flows out. If you do the naive approach and make simple channels like that, what will always happen is, as in this simulation here, you will have one channel which fills faster than the other because of small tolerances and cross sections, which basically means you start to trap a bubble here, and so this chamber doesn't fill. And to solve something like that, you can use indeed current simulation tools, but if you want to do a complete system simulation, there are no tools available for that. In addition, the devices are still very application specific. They are not the generic micro thing for assays or something like that. And there's comparatively little standardization. So how do you proceed? What we are doing is we try to do a 2-pronged approach simultaneously. On the one hand side, we do a very careful system level analysis, and we try to keep that system aspect always in the back of our minds. So we really try to understand the interaction of the various elements. In parallel, we try to do a bottom-up approach in the sense that we try to identify functional modules, we try to build them as a module, do module verification, and then try to add them up. So we try to develop and validate these modules for specific functions like sample prep, like lysis, DNA extraction, amplification, and so on. We try to use these modules similar to an element library in microelectronics, knowing, however, that there is that interaction. That's the system aspect of it, that there's always that interaction between these different modules. We combine them to run proof of concept experiments, and then we do a stepwise integration into an integrated micro device. So the strategic approach really is modularization here. And what we have, and I will show you an example, what we do there is, for many of these functionalities, we actually have off-the-shelf components, like the ones you see here, and which you can actually buy through a catalog. Things like, a module which does plasma or serum generation, mixers, chambers where you can do sample prep, amplification modules, and you can already see that they all have the same footprint. So these are sort of modularized or standardized to a certain extent, at least in their outer geometries. They have standardized interfaces like mini lures or lures here. The spacing of these pores are according to a 384 well plate, so again you can use a standard pipette to fill that stuff. So that's the sort of strategic approach to try to simplify this development. And during a development phase we try to combine these different modules, at the moment still in a chip in a lab situation. So you can really see this is sort of looks probably like most of what your setups look like in your labs. So we have a holding frame, we have these different modules here. For example, that one, that brownish stuff here are magnetic beads. So in this case we do sample prep, DNA isolation on magnetic beads. Here we have a chip which does continuous flow PCR for the amplification. Here we have a chip where we just have different chambers where we try to, in this case we lyophilize PCR master mixes, and we try out different geometries of these chambers to see which volumes work best, in which of these chambers the master mixes resolubilize best and fastest. And then we combine these, by having different tubings and connecting things together. And it's really like microfluidics Lego. But at that point, of course, it's still a chip in a lab situation. But once we have validated the functionalities here, we can start to do functional integration. And this is then how the end product really looks like. This is a true lab in a chip in the sense that it's a sample in, astro out device. You have a sample input here. You have a chamber here where we have this magnetic sample prep using magnetic beads. And the geometry of that chamber has been optimized by using these modules. We then aliquot the sample into 8 different streams. Here we have chambers where we have species-specific PCR master mixes lyophilized. And again, the geometry here has been optimized in these modules. We then do 8 parallel continuous flow PCRs. And then the amplicons go back on top of the chip in these channels to a detection zone. And again, the geometry of that detection zone was optimized by having one of these modules, and then everything goes off to waste. And then this is how it looks in the end. This is a single injection molded part. So you can make that for a few dollars. Has the size of a titer plate. It has a fairly high feature density. You can see that there are lots of features on this thing. But this is really industrially designed. So if we compare these, let's say the modular approach and the integrated device, during development we use this sort of Lego toolbox approach because it's very flexible. You can just exchange single modules and see what's happening. And the biggest advantage here is troubleshooting. During a protocol development, if you have an integrated device and it's not working, it's almost impossible to find out where the problem is, why it's not working. If you have these different modules, it's very easy to exchange the modules and to find out where the problem really is. The drawback, of course, is this is not a product yet, so this is a development platform. You have things like dead volumes coming from the connections here. And of course, if you add up the cost of each of these parts, it's a more expensive thing than something like this here, which of course is optimized for manufacturing. It has an optimized geometry. It does exactly one protocol, of course. So you lose all the flexibility here. This thing, of course, does only one thing. So this is the development stage, and this is then the final product. And we try to do actually the same also on the hardware platform. We try to modularize that as much as possible. Here for typical nucleic acid testing, so things like an arm with a magnet to move magnetic beads around, heating stages, fluorescence camera, or for immunoassays, which are typically more colorimetric based. So examples here. We have, for example, a point-of-care diagnostic system for celiac disease monitoring. So that's, again, it's the gluten intolerance. And in this case, for disease management, or let's say for diagnostics and disease management, you actually have 2 you would like to run two different assays in ideally one diagnostic system. So an HLA typing for the initial diagnostics and to obtain genetic information for the initial diagnostics, and then during the disease management you would like to have a serology assay, which is basically an immunoassay. And in this case we try to have a concept which has one instrument and two cartridges, but with a common footprint and interfaces. So the idea from the manufacturing point of view of having that standard footprint is, of course, you can use one common injection molding tool, one base plate for mold inserts, one assembly setup in the manufacturing, and one interface with the instrument. So everything in the manufacturing side becomes easier. And then you have to define what sort of interface you actually need. And once you define such an architecture, it actually helps you, again, from that system development perspective, to develop the instrument and the chip or the cartridge development in parallel. That's really important. And so what we do in such a situation is we try to define a cartridge landscape. Without really going into the functional details, we first try to identify certain areas of a chip to fulfill certain functions. So in this case, for example, fluidic interfaces, valve functionalities, heating zones where we say, in this case we would do an amplification in these areas here. Waste area, you can see, for example, the area we reserve for waste here is comparatively large. An area where we have sensors for detection. This again has the size of a titer plate. And then, of course, we start to build the individual designs based on that system architecture. And of course, as always, the devil is in the details. When we look at certain issues like, cost for liquid storage, for valving, for reagent storage, for example, in this case, PCR master mixes, compared to functional issues. For example, when we say liquid storage, what's the performance of the liquid storage? What's the shelf life? That's always a big thing. For the dry reagent storage, again, which reagents, how do we actually put them onto a cartridge? What's the shelf life? Or electrodes, how do we manufacture them? And then we come up with the actual devices. So that's the HLA chip, which is molecular diagnostics. So it has a PCR unit, so it has, again, something here for DNA extraction and metering. This is the PCR module. In this case, it's so-called shuffle PCR. So under the meanders, we have stationary temperature zones. So you don't do thermocycling, but it's a stationary PCR. And you push the liquid against this enclosed air volume, and then you release the pressure here, and the air is pushing back the liquid sample. So it's just going back and forth all the time, depending on how many cycles you need. And then in this case, we do an electrochemical detection of the amplicons. And if we look at the immunoassay chip, this basically has the same architecture, and again, fluid ports, different chambers, valves at the same locations, and again, detection at the same location. So in this case, we have one architecture and two different cartridges which follow the same footprint. Another example, point of care cartridge for the identification of bacteria causing urinary tract infections, because there we really have a clear clinical case in the sense that basically half of everyone on the globe will have a UTI at one point in their time. And actually in a substantial amount of cases, these UTIs become chronic, which basically means people have 3 to 5 chronic episodes every year, and it's fairly painful. In the acute care hospitals, about a third of all cases are UTIs. So it's really something which is very common. E. coli would be the typical cause for that. The problem is at the moment there are no good, fast methods for diagnosing that. So at the moment what you do for diagnosis is a bacterial culture from urine, which takes 2 to 3 days. On the other hand, if you come to a hospital and you have a chronic, you have an acute case of UTIs, they will just give you a broadband antibiotics. And of course we all know what's happening because of that broad and wide use of antibiotics. You start to develop resistance in bacteria, so you don't want to have that. So we started to come up with a fairly generic and simple design of a first generation integrated molecular diagnostic test for bacterial identification of UTIs. In this case we have inlets. We wanted to, in this case, process 2 samples. Inlets for 2 samples and 2 hybridization buffers. We have, in this case, chambers for stationary PCR, so conventional thermocycling. We then mix, after PCR, the sample with a hybridization buffer. We have a herringbone mixer here, and then we have channels where we do hybridization probe. We spot hybridization probes for the PCR products. That was the initial design, and this is sort of how the molded part looks like. Again, standardized footprint, microscopy slides, standardized interfaces. This is how the final fully assembled device looks like. We have two mechanical turning valves, and that's for the engineers of you. This, in my opinion, is the only working method for valves on a chip. I'm not a, let's say, big fan of PDMS. I don't think that PDMS is a material which lends itself for manufacturing, and despite the fact that some like 75% of all the academic papers in the last 15 years in microphysics has been on PDMS, with the exception of Fluidigm, nobody's using that commercially, and there's a good reason for that. So PDMS is not a good material. I always tell you, if you think about ever commercializing, or let's say ever get more out of your PhD thesis than a paper, don't use PDMS. That brownish stuff here are the lyophilized PCR master mixes in the PCR chambers. And these are just first results, and yes, amplification works. So what you see here are basically electropherograms of the PCR product for an E. coli strain. In this case, on chip with a liquid PCR mix, on chip with a lyophilized PCR mix, and these are just conventional thermocyclers, desktop thermocyclers. Works. However, we were not too happy with the PCR yield. So we came up with a second generation device, which is actually also moving closer to a real clinical product. So there are some improvements in the sense that we would like to run parallel PCRs together with a positive and a negative control. In this case, we move away from having two samples on a cartridge, but having one sample but two targets. So you can go for E. coli and the second most common bacterium. It turned out that urine has a certain PCR inhibiting property. So the initial concept was to go directly from urine, which, as I said, worked somehow, but not so good. So we now at the moment try to get rid of the urine. We also do a thermolysis in the protocol now. We do on-chip reagent storage of the buffers in blisters on the chip, and we then had to basically extend a little bit the footprint. So instead of having that very simple cartridge, we now have something which looks a little bit more complex. But I will talk you through the protocol in the next couple of slides. So this has the size of two slides, microscopy slides, so it's 75 by 50 millimeters. It has 4 turning valves, 3 blisters, and a couple of functionalities. So we'll go through the protocol. Step 1 is just to load the sample, and there's a membrane on which we then collect the bacteria, and we push the urine to waste. We then lyse everything on the filter with this lysis agent which sits here, and a thermal step. We then move the sample from the filter into a chamber which also acts as a bubble trap. We then load the negative control from that, let's say from the blister to the first PCR chamber. We load the positive control to the second PCR chamber. We load the sample aliquoted into chamber 3 and chamber 4. So we have negative control, positive control, and twice the sample here. And you can actually see how these different valves move when we do the different protocol steps. And that's the beauty of these mechanical rotary valves, that they are very versatile. You can use them for a lot of different functionalities. So you can see these have 8 ports, these have 6 ports, and always an internal, slightly different geometry. So this is, as I said, in our opinion, in our experience, the only really working valve technology on a chip. And this is how the cartridge really looks like. Final chip here with the blisters, the valves in the instrument. And we actually got that picture last week, so we haven't got any results on the protocol. We know that fluidically it's working, but I don't have any PCR results yet. And finally, the final example is a little on cell-based assays, because that's another big thing at the moment. So what if you have cells or particles, what can you do? With it, typically, first of all, you would like to separate stuff, like, for example, you have CTCs or other rare cells. There are a couple of ways of separating them, typically just by size, in a static way. So we have arrays of pillars. We have Weir filters, so with little slits on top of these diamonds here, so small particles squeeze through, big particles or cells are retained. Or we do everything on membranes, so we have one channel on top of the membrane and one on the bottom, and again, small particles can pass through the membrane and bigger ones not. We play around with surface functionalization to get nice long-term cell cultures in these chips. Again, it could be just on a hydrophilized polymer surface, or in this case on cell culture membranes in a chip. And then you can start to play around with different stains. Unfortunately, the contrast is not so high. And then you can start to evaluate all various cellular properties. To do a bit more complex cell-based stuff, we do organs on a chip, typically for drug discovery application. In this case, we're trying to build liver sinusoids on a chip, which is sort of the smallest functional unit in your liver is a sinusoid, which looks like this one here. So you have basically blood vessels here. Then you have a layer of endothelial cells, and in the middle you have a layer of hepatocytes. And we try to build exactly these structures in a microfluidic device. In this case, we assemble the cells using dielectrophoretic forces. So we have electrodes on either side of the channel, and depending on the field strength and the frequency, the cell types are assembled onto these ridges here. So again, in the middle you have hepatocytes sheathed basically by endothelial cells.
Wait, so I'm sorry, the dielectrophoresis concentrates the cells in the center?
Exactly. The idea is the cells move to the area of the highest field strength. And you can see that it has this three-dimensional structure, and by having this ridge here, you minimize the cross-section of the liquid between these two channels, and that way you create the area of the highest field strength just on the ridge, and that's exactly where you would like to have the cells to be. And that ridge is in addition coated with an extracellular matrix protein, so once the cells are there, they stick there. And so you basically apply an AC field with one frequency for the endothelial cells and with another frequency for the hepatocytes.
That's for toxicity testing?
That's at least let's say the first application. Yeah. Clearly, in the pharmaceutical industry, is toxicity testing. That's how the chip actually looks like. Again, standardized format. Here are these. We have 8 of these assembly units on each of the chips. The electrodes here, and here are these ridges on which we assemble the cells. This is a time-lapse image. So first, these green ones are hepatocytes, and so once they are assembled, we tune or we change the frequency of the field, and then we start to add the endothelial cells, which are labeled here with a red dye. And after a while, after they've been basically assembled here, we change the medium to a cell culture medium. And we've been running fairly long-term cell culture. I think the longest one at the moment is something like 2 weeks. The nice thing is you compare that three-dimensional cell co-culture to metabolic activity of just a flat 2D cell culture. You typically see almost 2 orders of magnitude higher metabolic activity. So that basically means the cells are, behaving much more in a in vivo-like fashion, like in a flat cell culture, and that's actually one of the most impressive pictures we've been able to take. You see these sort of little channels here. These are actually bile channels. These liver sinusoids actually start to secrete bile on the chip. This is something you never see in a flat cell culture. So these are really behaving in vivo-like.
What is that activity comparison to the in vivo? You gave me the 2D versus the chip, but what's the in vivo?
Good question. No idea. Would have to ask our biologists.
What are you measuring? Steroid production?
In this case, it's testosterone. Okay, so the last couple of minutes I will talk a little bit about, after we've seen all these strategic points and stuff like that, how do we actually make a business out of that? So one slide on the company. We're now in our 13th year, founded in 2002. We're a bit more than 75 people now, growing at about 20% a year. What differentiates us basically from all the other high-tech companies typically is that we're a family-owned company. No venture capital, no financial investors. Classically bootstrapped the company, growing on our own revenues. This is a building we moved in 2011. It's our own company building. We're just building the second building, decisive. Not going to all the technical details, something which from an academic point of view is totally irrelevant, but on the commercial side is really, really important, is you need quality control systems. So for ISO 9001, that's a standard one, and 13485 for medical devices. We are doing our devices in polymers, and that's the sort of technology chain you need for manufacturing polymer-based micro devices. Starts off obviously with designing microfluidic devices, but as I said before, already at that point, you have to get all the different aspects, the multidisciplinary aspects of it into the process. If you miss something like that, it will not work. So application know-how is really important, and we indulge in the luxury as a manufacturing company that we have cell culture labs, nucleic acid labs, immunoassay labs where we work with clinical samples. So I always joke we're the only cell biology lab with injection molding machines, or on the other hand, we're the only injection molders with real-time PCR machines. Physics, microfluidics, material science, and manufacturability. If you're not, at that point in time, very, very early in the process, build manufacturability in the design, you will never hit your price point. And I elucidate that in a couple of minutes. So once we have a design, because we're talking about polymer replication technologies, the next step is to make a master structure, the geometrical inverse of your device. And you can throw the whole range, the whole plethora of microfabrication technologies at that problem. What we have in-house and what we like to use most are the first two here, ultra-precision mechanical machining or electroplating. Ultra-precision mechanical machining has the beauty that it allows you two things. One is a large dynamic range in structural dimensions. So you can make a 50 micron channel besides a 1 millimeter high structure. It also allows you three-dimensional structures like ramps or funnels or stuff like that, which would be very difficult to make with other microfabrication technologies. Limiting factor is the radius of curvature of intersecting features. The reason for that is fairly simple. If you make a channel in a microfluidic device, in your master structure, of course, that is a ridge. And to create a ridge, you have to machine material away from that ridge, which, as long as you make a straight channel, you can make it as narrow as you like. But if you have two intersecting channels, then of course you have to remove the material out of that corner here. And if you have a rotating mill bit, you can obviously only create a radius of curvature which is as small as the radius of curvature or the radius of your smallest machine bit. And for any practical feature, that radius is about 25 micron. Which actually it turns out for most real-world applications that's good enough. If you need sharper edges than that, then we would be going back to classical lithographical technologies. We use either a resist structure or a silicon structure, which is lithographically defined, and we would electroplate that. Drawback there is then you lose that dynamic range of structures, because in lithography you can make things, you can make a 2 or 3 mask lithography, but then one layer might be 20 micron high, the other one 50 or 100. But that's a sort of factor of 3 to 4, which you have as a dynamic range, and not 10 or 50 or something like that.
Well, do you need that dynamic range generally?
Yes. That would be a good example. Look at that waste reservoir, and there are 100 micron channel on this thing as well. So you would like to be able to do all that with a single technology, or something like this. Just fluidic ports. That typically is a macroscopic feature. So we see a huge demand of dynamic range on devices. Once we have the master structure, we would replicate that into polymers. And again, you can throw all different kinds of replication technologies at the problem. 97% of what we do is injection molding, because that is a nicely scaling technology, which once you start, you can make thousands or millions of parts. Thermoforming is another technology. You know all that. This is a classical example of a thermoform part. Thin wall structures, yogurt cups, stuff like that. We mainly use that to do blister packs for liquid reagent storage, because the replication accuracy is really bad. You can't really make a 50 or 100 micron channel with thermoforming, but a macroscopic thing like a blister is good. Hot embossing, we have a hot embossing machine, because historically that has been the first technology to make microfluidic devices in thermoplastic material. And from the academic point of view, much rather than going to PDMS, I would invest $50,000 on a Tauber hot press or something like that, and do hot embossing in thermoplastic materials. The big advantage here is the replication accuracy. If you want to make a 50 nanometer channel, hot embossing is the way to go. Drawback is the cycle time in hot embossing is comparatively long, typically several minutes, so in terms of throughput, that is limited. Then of course, famous PDMS casting. We always try to talk people out of that. It's not a good manufacturing technology. Once in a while, people come to us and want to have a few parts, so instead of, carrying their money elsewhere, we take it, of course, but we try to talk people out of PDMS. It's not a good material for industrial processing.
Which is the most limiting property of PDMS in your opinion?
There's not only one. It's expensive. PDMS is $100 a kilo. Polycarbonate is $6 a kilo. High performance polymers like COC or COP is $13 a kilo. Even if you do reactive injection molding with PDMS, the cycle time is still several minutes. If you do it on the conventional way of pouring it and something like that, even if you put it in the oven, it's still an hour. So throughput is limited. PDMS is a spongy material. So if you have an application where you have small molecules, you have to do surface coatings because otherwise your small molecules just disappear into the polymer matrix. Reproducibility, I mean, you have to control the properties of your environment extremely closely. I mean, PDMS is, I think properties of PDMS very often depend on the phase of the moon or something like that, so it's very difficult to really get a highly reproducible protocol. And I think the yields in Singapore are still fairly poor from what I hear. So, yeah, it's just not a good material for volume production. And of course, the mechanical, interfacing it is a pain, so that's why you have to typically have to make a cassette around it in some case. So, yeah, it's not a good material. At that point, you have a structured polymer part, and to make a functioning microfluidic system out of it or a device out of it, you have to go through all these back-end processing steps. On the left-hand side, all the physical steps like cutting, sealing, assembly. That's something which is becoming more and more important because we see an increasing heterogeneity in materials. So instead of just having an all polymer or all glass device, you have something which has membranes and filters and septa and electrodes and sensors and whatever in there. So assembly is a big one, and metallization to put electrodes in the device. And on the right-hand side, you have all the more chemistry-related issues like immobilization of biomolecules, the reagent storage, the tuning of surface energies and stuff like that. And then finally, quality control, which again in industry is a huge thing. Typically, it's a two-stage process. We do an optical inspection and then we do functional tests. And I can tell you that we have a product out on the market where the client wants to have 100% functional test of a disposable, which took us about a year to come up with a testing method which does a non-destructive testing of a disposable. But they want, it's for the US military, and we do that in some 10,000 parts a year. And for that product, the QC cost is 80% of the overall manufacturing cost. It's sort of, you get what you pay for. What I would like to draw your attention to is that in the academic world and also during the early stages of the development, people typically concentrate on that area here because that's where for low volume manufacturing 80% of the cost is. In volume manufacturing, however, 80% is in the back end. And that's something where people very often during the development don't put any, or let's say they don't have that on the radar. And that's another reason why a design to manufacture is so important, because the money saving potential is basically here. It's not here. So it doesn't actually make any big difference if you shave off 5 seconds your injection molding cycle. That has no big effect on manufacturing costs. However, if you can simplify your back-end processing, or if you can even eradicate one back-end processing step, that's where the money-saving potential is. But that has to be designed into the device. So if during the design phase you miss that bit, you're locked into a situation where your money-saving potential is, or your economy of scale is severely limited. So that's really important to concentrate on back-end processing. Okay, a few words on our business model. ChipShop basically is active in 4 fields. We have off-the-shelf products, we do complete systems, we do customer-specific development, and we do collaborative R&D projects. The off-the-shelf products, the catalog, that's exactly the reason I explained before, to lower the entry barrier for using microfluidics. So now, for these early proof of concept stages, an engineer can simply, buy a couple of chips from the catalog out of his own material budget and run a proof of concept experiment. He orders the stuff, a few days later it's on their desk, they can just try it out. And if it doesn't work, he has spent $50 or $100 and not 50,000. That's the big difference. The second one is, as I said, the biggest part of our business, of course, is customer-specific development and manufacturing. And my mantra is use scalable technologies right from the beginning, also for prototyping. Because if you do your prototypes with manufacturing method A, like say laser cut laminates or PDMS, and then at one point would like to scale up, you have a problem because at one point it's not a seamless transition. And so for us the worst situation is somebody comes with a PDMS device or a laser cut laminate stack, and then now I need 10,000 of these or 100,000 of these. It's basically back to the drawing board because the results of the prototypes obtained with manufacturing method A and material A will not scale or can't be translated one-to-one into something which is made with another material in manufacturing method. What we are doing is and that's the other reason for the standardization, we use so-called mold bases or standardized injection molding tools. That's an image of such a molding tool, and something like that can easily cost $100,000 or $150,000. So that's where the image from injection molding is really expensive comes from. But if you have a modular tool where during the development phase you work with standardized mold inserts, so that's the reason for the standardization. So you only have to pay during the development cycle for this part here, for a mold insert with your specific structure. Then injection molding also suddenly becomes price-wise comparable to other prototyping methods. The advantage is once you have that mold insert, shooting 100 parts or 1,000 parts or 10,000 parts, doesn't matter. And so you have that seamless upscaling potential. And what I always tell people, it's at the moment, once you have that, it's easier for us to make 1 million chips than for you to sell 1 million chips, saying that manufacturing capacity typically is not the bottleneck, it's the market. And the same we've done for all the back-end processing steps. I told you that 80% of the manufacturing cost is in the back-end, so we put a lot of effort into coming up with scalable solutions for liquid and dry reagent storage, for things like surface modification, for spotting of biomolecules, for the integration of membranes here, or septa here, of electrodes, or these mechanical turning valves. And in addition to manufacturing, we have the ability to do really the functional validation of what we're doing. So that's what I mentioned, that we have all these biolabs where we can actually really validate what we're doing and don't have to send the stuff off to somebody else to do the validation.
During the prototyping phase, the design can change like on a weekly basis. Do you think it's worth it to make a mold?
Yes.
From you guys?
Yes. The story I tell there typically is when people really only want to have 5 parts, we can do, we would do direct machining into one of our molded platforms. So we have a molded part which has all the fluidic ports and stuff like that, but a blank backside, and we just machine everything directly with an ultra precision machining tool into this side. However, that is a serial process. That takes a couple of hours for each of the parts. So let's say you want to have the first 5 parts, and together with a little bit of CAD work on our side, that might be $3,000 to $4,000 for the first couple of parts. We make these 5 parts, we ship them off to the client. A week later the phone rings and they say, yeah, we've tested the parts, they work really nicely, but of course, you go through 5 parts like nothing, so we need another 5 parts. Because it's a serial process, the next 5 parts cost maybe not $4,000 but $3,000. A week later, the phone rings again. Oh, colleagues have seen these parts and they have a couple of crazy ideas what to do with them. We need another 10 parts. So you end up with some like $10,000 to $15,000 for, at that point, maybe 20 parts, which is about the same as making actually a mold insert and doing 100 parts. And if you do a real product development, you would like to do statistics. You need, let's say the larger diagnostic companies order 5,000 to 10,000 parts for their in-house testing. So it doesn't, don't start with 5 or 10 parts. It doesn't make any sense. And by now our cycle times for as long as you can work with these standard modules is about 4 weeks. So as long as we can churn out parts faster than the time it takes the clients to validate what they're doing, we're fine. And actually, it makes a lot of sense to maybe think a little bit more carefully on the design than just, via trial and error go through many designs in, with 2 chips, or 5 chips or something like that, because it's not really clear what you learn from 5 chips. The third part of the business are complete systems, and that's actually a field which is growing quite rapidly, because we've made the experience that it's very, very important to very quickly enable people to also do their assay development on a microfluidic basis. For this reason, we do breadboard instruments or little bits and pieces like this one here. I mean, we're not primarily an instrumentation company, but we can make these little instruments like this one here, which does magnetic bead-based sample prep for molecular diagnostics. So we have a moving magnet here which pulls magnetic beads through a chip, and a heating foil underneath, so you can raise temperatures to about 75 degrees for thermal assisted lysis. We have a universal instrument, a universal thermal controller for all kinds of chip-based PCRs. We have the microfluidic system, and when we talk about complete systems, it's an instrument, it's chips or a family of chips, and it's everything around. So pumps and flow controllers, then all these little support kits. I mean, there's nothing more annoying than spending $30,000 on your latest toy, and then there's a 10-centimeter piece of tube missing. So, and application protocols. As I said, we indulge in the luxury of having an application group. So we typically provide people also with an assay coming with their specific design. Again, I would not call us an assay development company, but for sure we're an assay adoption company. So very often we have clients which give us an existing assay which say runs in a 96 well plate or an Eppendorf or something like that, and we put that onto a cartridge. And finally, collaborating R&D projects. This simply has to do with the fact that microfluidics is still a moving field. It's scientifically evolving, and we try to keep at the cutting edge there. So as long as we get my papers accepted at µTAS or so, I'm happy. More important from the company point of view is, of course, the fact that we're still a small company, even with 75 people. So for us, it's important to create these networks of excellence. We have to know who knows what, and ideally just be able to call them up on a short notice and get an answer to a problem. Or if you can solve certain aspects of a commercial development in the framework of a publicly funded project, that of course helps to save money. So that's, we have a fairly, let's say we're still fairly R&D heavy in that respect. Okay, so coming to conclusions. Microfluidics at the moment, we really experience a dramatic increase in commercialization. So finally, after these 25 years, we see things going, and it's also quite nice. I've been reviewing projects for a couple of funding agencies, and for years they've been pressing and saying, no, we've been pouring money into that field for 20 years now. When do we see some taxpayers pay back? And I think we're at that point in time by now. The advances of microfluidics technology really make the development of these complex microfluidic devices possible, which is really fulfilling one of the early promises. This sort of, sample in, drink coffee, result out situation. The complexity of the protocols we put on the cartridge is really rapidly increasing. I mean, we now do cartridges which, 5 years ago I was like, whoa, this is never going to work. So this is pretty good. What is really important in the development of point of care or in general these microsystems is to do a parallel development of a microfluidic cartridge, a chip, the instrument, and the assay. It's always a bad situation if one of these three is either too far advanced or is lagging too far behind. And this goes back to this sort of holistic system approach I mentioned a couple of times. In our experience, modularization is really a key for a fast product development, and we modularize the cartridge, the instrument, and ideally also the assay. I mean, we have certain assay steps validated on modules, which you can, again, also put together in this modular fashion. However, the development still follows a device-specific or application-specific path. So it's, even if we have these modules, we always have to tweak them a little bit for that specific application. And again, to make things easier and faster, try to use existing standards wherever possible. I think one of the reasons why in the early days of the first commercial devices took so long to get accepted were that they were all standalone systems. They did not work with what was out there in the lab. I think Caliper made a huge strategic error to come up with a system which was just sitting there and had no interface to existing lab infrastructure. And so that's why we work with these geometrical standards, standard port interfaces and stuff like that. So I think it's a good time at the moment now for the academic and for any professional community to enlarge their tool set. So to get away from Maslow's law of the instrument, so you still need hammers, but you would like to have many, many more different tools. I have to acknowledge a couple of funding agencies because quite a bit of the work I was able to show you have been publicly funded. And finally, of course, my last slide, I think we're all fairly familiar with the concept of modularity, and what works well in the child room might also work well in the development of micro devices. With this, I'm at the end. I'm, of course, happy to answer any questions, and thank you for your attention.
Can you point to, like, I guess a critical mistake that led to a really, really good learning from all these years?
You mean there is some sort of element of serendipity in, when I can't? Yeah. But I let's say, nobody of course talks about that. You always see the end results. So, let me try to think if something pops up in my mind, but not really. I can't really think of something where, you have this totally accidental discovery or something like that in microfluidics. I don't think so. I mean, that's actually quite interesting in the sense that if you look at the physics of microfluidics, many of the phenomena you see, actually all the way down to almost nanofluidic things, is comparatively intuitive. It's not like quantum mechanics where suddenly you start with these things which are totally counterintuitive. Many of the things here, I mean, people have for a long time been using simulation tools, and what I've seen as results there in most cases was that, my gut feeling tells me it should work like that. So I don't think that, I think there's comparatively little room for this sort of serendipitous discoveries.
I was wondering if you could please elaborate a little bit on the challenges of the waste disposal part, especially with potentially infectious material, and I don't actually know what typically people would do. Would they incinerate those things and affect the materials?
Yeah. I mean, it's basically, if you have a disposal in a hospital, and that's regardless if there is an infectious sample or not, this is clinical waste. So that has to be disposed of as clinical waste. What you would, of course, like to do is that any liquid which goes into the cartridge should stay on the cartridge. And that basically means you typically have fairly large somethings in the cartridge. What you also have to take care of is, of course, when you try to fill a big reservoir, the air has to go somewhere. So typically these reservoirs have a hydrophobic patch. Let's say they have a venting outlet which is sealed with a hydrophobic patch, so the air can go out but the liquid stays in. As I said, the challenge typically is just the sheer volume of that thing. For the molecular assays, it's often not so critical, but for an immunoassay, when you have different wash steps, you can very easily come up with, maybe I don't have it with me, but I can show you a picture here in the catalog of such an immunoassay device, where you can see that has a huge waste reservoir. I mean, you can see like this one here, That's, I think, 2 milliliters. I mean, for microfluidics, that's a huge volume, and that just comes from the fact that you need 3 or 4 different washing steps.
And what if you were using something in, say, a remote setting in Uganda and you're dealing with Marburg virus or something?
Yeah.
Do you ever have clients who, clients who have some special waste disposal requirements, or?
Not so. I mean, incineration is actually the way to go.
In terms of market adoption, can you give me some sort of rules of thumb in terms of costs? I mean, if you're making 10,000 units and you think you want to get into a market and you think microfluidics is the way to go, what should I expect when I'm all said and done? Kind of what that running cost is that device that, I mean, obviously there's a scale issue, so I don't know what that, I don't know where to start with that, but maybe you have a rule of thumb.
Yeah. The rule of thumb for scaling is if your device, let's say, is of medium complexity and you don't have too many assembly steps because they don't scale so well, is a factor of 10 in production volume leads to a reduction of 2 in price. So And that actually holds quite well. So let's say we make a small series of 1,000 parts, and something the size of a microscopy slide, as I said, with average complexity, maybe with a filter on it or something like that, at 1,000 parts will, as manufacturing cost, will be maybe something like $10 at
Per
1,000
unit.
parts per unit.
Mm-hmm.
At 10,000 parts, it would be $5. At 100,000 parts, it would be somewhere between $2 and $3. And at 1,000,000 parts, you would hit roughly the magic dollar mark. And that rule actually holds comparatively well. I mean, within plus minus 50% or something, but that actually holds quite well. And the higher your production volume becomes, the more critical the influence of the raw material cost becomes. So let's say we take Zeonor, a part of like that has a shock weight of maybe 5 to 7 grams or something like that. So the raw material cost here is of the order of maybe 15 cents. Which, you know, if you do 1,000 parts at $10, the 15 cents is a percent. So you don't really care. If you go to 1,000,000 parts, the 15 cents suddenly becomes 15% of the manufacturing cost. So if you switch from Zeonor to a polycarbonate, you suddenly save 10% of your manufacturing cost. So the role of the material cost becomes more important the higher the volume becomes. And then again, the next thing is, as I said, assembly doesn't scale so well. So that's another reason for that. How do I design my part initially that can be manufactured? If you can simplify that assembly, or if you can design the part that you actually need lesser number of parts which you have to assemble, that's where the cost saving potential is. Because certain processes don't scale so well. But in general, it's a factor of 10 in production up, a factor of 2 in price down. That's a fairly actually good rule of thumb.
So in developing the immunoassay on lab chip, to what extent does your group have expertise with reagent chemistries? Do you utilize mostly your collaborators' expertise for that, for localization of reagents, things like that?
I would say it's half-half. It's surprising that, let's say, I'm a big fan of Pareto's law. So with 20% of your whatever money, knowledge, or whatever, you can solve 80% of the problems. And so by having that sort of certain basic set of protocols and reagents, you can actually solve 80% of the application cases just by slightly tuning them. And so we were quite surprised, actually, a couple of years ago, we were working for a fairly famous, let's say for a diagnostic company which is fairly famous for their sample prep protocols. And they were asking us to basically take one of their existing protocols and put it on a cartridge. And they had tried a little bit in-house. But we were capable of, within 4 weeks or so, come up with a sample prep protocol based on their existing chemistry, but just, what you typically do if you scale something down, you have to change the ratio between different reagents because surface to volume ratio changes. Or typically if you have a macroscopic assay, people are using wash liquids in excess. If you scale things down, it turns out that you don't have to have that factor of 5 more wash buffer in that step compared to a macroscopic assay, but a factor of 2 is enough. So the ratio between a certain reagent and, say, wash buffers or so is changing if you put it on a chip. And that's something we're pretty good and simply based on our experiences. Because, as I said, we have maybe only made 20% of the cases, but with these 20% of the cases, we can solve 80% of the application cases. Okay, I think time to go home, huh?